Okay, so I've changed the title. The slides will be in French, so I will speak in English. Because I'm not a specialist of RNA virus, by the way, and when Philippe asked me to give a presentation for this opportunity, I always like to come to the EHU and give a talk. So I think, what can I say about RNA viruses? Maybe I can come back to my older hypothesis that this RNA virus play a major role in the origin of DNA, and try to have an update with what we know today about RNA virus. And the question will be, did a modern DNA virus originated from RNA viruses? So briefly to remind the main difference between RNA and DNA, basically DNA is a modified form of RNA. That is the important point, with two major modifications: the removal of the oxygen in 2' of the ribose and the transformation of the uridine in thymidine by methylation. And these two differences are very important because the first one, the removal of the oxygen, makes the DNA a much more stable molecule than RNA, because this oxygen can attack the phosphodiester bond. And this oxygen, which is very important in RNA for the ribosome activity, for example, is also the dark side of this oxygen is that it makes the RNA unstable. So removal of this oxygen makes DNA more stable. And of course, also a very important point, if you'd have an thymidine instead of uracil, because deamination of cytosine is a spontaneous chemical reaction which transforms C into U. In RNA, you cannot discriminate a good U and a bad U which has been introduced by a spontaneous deamination of cytosine. In DNA, the cell can recognize the dark duck because normally you don't have U in DNA. So when you ask why RNA has been replaced by DNA in the course of evolution as a genetic material, people used to say, okay, it's obvious because DNA is more stable than RNA. It can also be replicated more faithfully. And this is very important to increase the size of the genome. In fact, to have big genome like the human genome, you need to have DNA and not RNA. So usually people stop there and say, okay, so it's obvious DNA replaces RNA. But then I used to say only God could have predicted that in order to have the big human genome, you need at some point to replace RNA by DNA, or maybe intelligent design. So this kind of reasoning would be to say that why dinosaurs had feathers, just because birds need feathers to fly. So you have it was nice for dinosaurs to have already feather to be ready to become birds. Of course, evolution doesn't went like that, and this is a slide. Didier can maybe don't look at this one. What Darwin asked is, what was the selective advantage of the first organism with an RNA DNA genome? And at the time of the transition between RNA and DNA genome, probably the genome were very small, so it was not a big advantage to become. The genome cannot become larger at once very rapidly. And of course, there was no mechanism of repair to recognize the presence of uracil in DNA instead of thymidine. So this advantage to be able to correct cytosine deamination could not be useful at that time. And because we have examples also of viruses which modify chemically their genome, there are many DNA viruses which are known to modify their genome. This was the first well-known example in the case of T4, where all cytosine has been replaced by a hydroxymethyl cytosine, or even sometimes there are even more complex bases. For the viruses, it's very useful to chemically modify their genome to become resistant to the enzyme of the host which attacks this genome. So with this reasoning, I suggest that maybe DNA was first invented by viruses, or selected in viruses as a modified form of the genome to protect them against the RNAs, for example, of the host. And I published this idea in 2002. And for me, there were several arguments in favor of this hypothesis. The fact that many of the enzymes which are important for the transition between RNA and DNA, ribonucleotide reductase, thymidylate synthase, reverse transcriptase, and so on. Many of them are encoded by viruses, and usually they are also very divergent in viruses compared to cells. They have a huge variety of these enzymes. So it makes sense that this enzyme first originated in the viral sphere. And now in modern cells, the precursors of DNA are made from the precursor of RNA at the level of the monophosphate. So you have the ribonucleotide monophosphate, which are transformed deoxyribonucleotide monophosphate with DUMP at some point, and DUMP is transformed in DTMP by the thymidylate synthase. So the idea was that RNA viruses were first transformed into DNA viruses with the invention of ribonucleotide reductase, then tDNA viruses with the thymidylate synthase, and at some point the DNA was transferred from virus to cell. So one thing which is known now, and this is a slide I picked up from Eugene Kounin, who showed it recently also here. We know now that all the enzymes which are involved in this transition and in the replication of RNA and DNA genome somehow seems to have a common origin. It's possible to find in all these enzymes the fold which has been called the RNA recognition motif, which suggests that in fact all these enzymes evolved from ancestral proteins which were present at the time of the RNA genome. So this is one thing which can be in line with the hypothesis. And for me, this hypothesis was also strongly supported by the fact that the viruses exhibit a huge diversity of enzymes involved in their DNA replication. In fact, the diversity of enzymes involved in DNA replication is much larger in viruses than in cells. And it seems that the DNA replication mechanism in archaea and eukaryotes on one side, and DNA replication mechanism in bacteria, and they are not homologous, they are completely different. They seems to be a subset of all the DNA replication mechanisms which are present in the viral sphere. So a bit like the out of Africa hypothesis. So the gene from the Eurasian people are a subset of genes which are present in Africa. I suggested maybe there is an out of viruses hypothesis for the origin of DNA. And in this model, of course, in fact, you have this transition from RNA virus, uDNA viruses, and in fact, some viruses are known to have a genome made of DNA with uracil still, uracil and not thymidine. And then virus with DNA genome, normal DNA with thymidine. And we know that there are two enzymes completely different, not homologous, TIA and TIX, which are able to transfer DUMP in DTMP. So in fact, DNA, as we know it today, has been invented twice independently by the origin of these two enzymes. One of them has been discovered in my lab in Orsay in 2000 by brilliant postdoc Anu Milikallio. And of course, in this hypothesis, the obvious thing is to think that, okay, DNA viruses originated from RNA viruses. And during this transition, they were transferred to cell. But is it true that DNA viruses originated from RNA viruses? In 2002, when I proposed this hypothesis, we knew nothing about the relationship between the major groups of viruses. So we have no idea of the evolutionary relationship between RNA and DNA virus, not even between DNA virus themselves. The situation has completely changed the last 20 years, and even more the last 10 years. With the advance in structural genomics, now we know the folds of many capsid protein of many viruses, and some unexpected similarity were detected between viruses which were supposed to be very divergent. And we also know the structure of reverse transcriptase, for example, RNA replicase of RNA virus, which seems to be homologous. And based on this knowledge, recently the ICTV are proposed to completely reorganize the classification of viruses, and Mart Kripovich and Eugene Kounin have been instrumental in this process. Now the viruses have been grouped into 15 level, taxonomic level, so this is a bit too much in my opinion, based on their capsid protein and their replication protein. So last time when I come here and discuss about this classification of viruses, I say that the term realm in English, which has been used by Koonin to name the higher taxonomic rank of viruses, was translated in French by domain. And the idea of to speak about realm and not domain was precisely to distinguish viruses from the cells. Now I realize that in fact it was possible also to translate realm by sphere in French. So now I use sphere in that case. And what we know now is that in fact, yes, this sphere groups viruses which are supposed to be related, evolutionarily related, either by their capsid protein or by their replication protein. So viruses from different spheres, in contrast, seems to be completely unrelated. So this indicates that viruses are polyphyletic. We cannot determine the LUCA of viruses, the common ancestor of all viruses. And so now we have already different spheres which have been proposed by the ICTV, more coming. And important point is that DNA and RNA virus are in different spheres. So it doesn't seem to be really evolutionarily related, with a single exception with the Epidnaviridae, which are obviously related to retroviruses and viruses encoding reverse transcriptase. So in blue, you have RNA viruses. In red, DNA viruses. You see that all RNA viruses are grouped in a single sphere, the riboviria in single realm, except for one RNA viruses that have put it an entire sphere for this single virus related to viroid. So in this analysis, we cannot say that DNA virus came from RNA viruses. So as I say, all RNA viruses have been put in a single sphere called riboviria because they have all homologous RNA, dependent RNA polymerase. And these RNA-dependent RNA polymerase, they are very, very divergent in the different family, but they are homologous, and they are distantly homologous also to the reverse transcriptase. This riboviria realm has been divided in two main kingdoms, ortho-RNaviria and para-RNaviria. These are rather simple to grasp because the ortho-RNaviria correspond to the viruses which encode an RNA-dependent RNA replicase, like the coronaviruses. And the para-RNaviria groups all enzymes which encode for a reverse transcriptase. And then you have subgroups, different phylum, with very strange names sometimes. I didn't try yet to memorize all of them. In some cases, rather easy, like we have duplo RNA viricota, they are all double-strand RNA viruses, or the nega RNA viricota, they are negative sense RNA viruses. But you see the three other ones, you have both double-strand and single-strand RNA virus, or you have positive single-strand RNA virus in two different phyla. In fact, this phyla has been defined based on the work by Koonin and his co-worker, many of co-workers who published a few years ago a phylogeny based on the RNA replicase of these RNA viruses. And in this paper, this phylogeny has been criticized by others because, in fact, the sequence similarity between all these different RNA replicases are sometimes very low. But they say they succeed to convince the ICTV. And you see that here you have 5 branches, and each of these branches correspond now to one of these phylum I mentioned previously. Most of these RNA viruses are single-strand RNA viruses. At least the single-strand RNA viruses are the most abundant one. And many of them have the same type of capsid fold, which is called a single jelly roll fold. But there are diversity of the fold in some of them. Yes. Most of these viruses infect eukaryote. In fact, we don't know any RNA virus infecting archaea, very few RNA virus infecting bacteria. But in their phylogeny, here, you see that they root the tree using group 2 intron and retrotransposon. So they root the tree with DNA mobile elements present in bacteria. But this is arbitrary, by the way. And the few RNA viruses which infect bacteria are mainly at the bottom of the tree, the Livividae, and a few also, most at the top, the Cystoviridae, which clearly came from RNA viruses infecting eukaryotes. So, in fact, in the tree, it's like if all these eukaryotic RNA viruses came from bacteria, which is a bit strange for me. They publish also a phylogeny of the virus encoding the reverse transcriptase, and in that case too, you have as an old group the mobile element present in bacteria. Here you see that DNA viruses, in fact, seems to come from RNA virus in this subgroup, but clearly you cannot say that they are at the origin of other DNA viruses, so there is still no connection. In fact, why Koonin and also Mark put the tree this way with bacteria at the base, it's because they are strong proponents of the doxa now of the tree of life, in which eukaryotes came from the fusion of bacteria and archaea. And this creates a problem for the RNA virus, because most of the RNA viruses here in red are in eukaryote. There are no RNA virus in archaea, very, very few in bacteria. So this raises a major evolutionary question: what is the origin of all these RNA viruses? For Koonin, they all come from these small RNA viruses present in bacteria. Personally, and with my friend Violette and Morgan, we think that this so-called 2D model in which eukaryote came from by the mixture of archaea and bacteria, and especially at the moment people believe that this eukaryote at this site came from the subgroup of archaea called Asgard. We have shown in this recent review and analysis that there are many biases which can explain this 2D tree, and in fact one cannot reject the 3D tree in which all the three domains are monophyletic. I will talk about this next time when I come here. So if that, in the case of the three-domain tree, it's possible to imagine that, for instance, RNA viruses are much more ancient. They were already present at the time of the RNA cell, when it seems logical to think that when cell had an RNA genome, they were infected by RNA viruses. And you can imagine, for example, that they are still there in eukaryote, but that they have disappeared in archaea. Maybe because the ancestor of archaea was a hyperthermophile, and I tell you that RNA is very fragile at high temperature. So one hypothesis is that archaea succeed to get rid of all RNA viruses. And some works have shown that maybe bacteria also have some thermophilic ancestors, or maybe also bacteria succeed to get rid of not all of them, but some RNA virus. So there are two alternative possibilities now which are in discussion. And with Morgan, we suggest that maybe, in fact, we can root the tree of Koonin differently in the middle of all these viruses which have single-strand RNA viruses with a jelly roll fold. And in that case, you can imagine that these RNA viruses are more ancient and came from the time of even before Luca. But still, there is no, at the moment, obvious evolutionary link between the genome of RNA virus and DNA viruses. And recently, genomic analysis has been published by Kunin and his co-workers. They did a metagenomic analysis and they found they double the number of known RNA viruses, and still all these new RNA viruses came into the already defined branches and families. So no new, completely new RNA viruses. No big, the coronaviruses are also still the largest known RNA viruses. Yes, I will. There is one case interesting is that this group, this sphere of DNA viruses, mono-DNAviria, which are most of them are single-strand or double-strand, came from the combination of a bacterial plasmid and capsid of an RNA viruses. So there are sometimes some connection, but not in the sense that I used to consider. So I really try hard to see how I can end up this talk, because at the end I don't have any argument to support or for or against my hypothesis. One can imagine, for example, that in fact DNA was not invented by viruses themselves, but by cells in the fight with viruses. So you can have which is more in line with the idea of Kunin and Krupovich. My conclusion is that in fact the only strong point for me is probably the transition between RNA and DNA really needs this reverse transcriptase. And all known reverse transcriptase are homologous. I don't know if we will find someday a completely different reverse transcriptase. So it's likely that this the reverse transcriptase we know is the one which was involved in the transition. So I look for phylogeny of the reverse transcriptase, and in fact I don't find recent phylogeny. I found this phylogeny from Irina Arkhipova, who is probably the woman, the scientist who has best knowledge about this reverse transcriptase. And the only thing I found interesting is that if we look at the distribution in eukaryote in red and bacteria in green, we can see that this enzyme is widely present in the bacteria with very different form in viruses, in other mobile elements, and only present in a few bacteria, and to my knowledge at the moment not in archaea. So there is a kind of correlation with what happened in the case of RNA viruses. There is a kind of overlapping between the distribution of RNA virus which use an RNA replicase and this RNA virus or DNA virus which use a reverse transcriptase. So I end up here without real conclusion, and I just want to thank Philippe and Didier for this invitation.